In this lecture, you will learn about the development of the human microbiome, in particular the gut microbiome. How did microbes get into our guts? Do we get them from our mothers or from the environment? Do they change over time and become more complex like the immune system does? In this image, we show a healthy human adult female and highlight areas that we know host a distinct microbial community. Arrows illustrate potential ways microbes may disperse, invade, or change in different body sites. As we learned in the last lecture, when we sequence the microbial genes, otherwise known as the microbiome, in and on in a human adult's body, we find that different sites on the body have unique microbiomes. In our data plot here, each point represents a microbial community from a healthy woman's mouth, skin, vagina, or gut. To sample the microbes of the gut, we actually sample fecal material, which is a really good representation of the microbial communities that are found in a person's large intestines. On this plot, the distance between points represents how different the microbiome of samples are. We will talk more about these types of plots in week 3. The important trend to understand is that microbial communities are distinct in different body sites. For example, if you sample 3 healthy adult females, the mouth microbial communities of each person will be more similar to each other than a mouth and gut sample from the same person. When a baby is born, does he or she have a similar set of distinct microbes in each body site? The answer is no. In fact, it doesn't matter where you swab a newborn, his or her microbes are similar across all body sites. Unlike adults, their microbial communities have not developed. So where do these first microbes come from? Well, it depends how the baby enters the world. If a baby is born through the birth canal, the baby's first microbes will primarily come from his or her mother's vaginal microbiome. Here we show 3 microbial communities sampled from the baby, and they all cluster with the vaginal microbiome samples. A healthy woman's vaginal microbiota primarily consists of lactobacillus, which is a genus of bacteria containing many species that are important for human health. So what happens if a newborn is delivered by C-section? Instead of a microbiome resembling the vaginal community, the baby's first microbes will look more like those found on the human skin. Here we show 3 microbial communities sampled from the baby, and they cluster with the skin microbiome samples. Are there health implications for infants with a more skin-like microbiome? We know that cesarean births are also associated with higher rates of a broad range of diseases, including asthma and food allergies. Could some of these issues be attributed to microbes? We don't know, but it's an active area of research. For mothers who must have C-sections, are there options to expose their newborns to microbes from the vaginal community? Well, Professor Maria Gloria Dominguez-Bello of NYU has been studying this exact question. We will hear her talk about her research in an upcoming interview. Another important question about the development of the human microbiome is, what happens to an infant's gut as he or she grows up? When does a child's gut look like an adult gut? Let's look at some real data. Here I show the data from the Human Microbiome Project. Microbial communities differ greatly between different body sites, as we learned in earlier slides. We do see that the vaginal microbiome is similar to the skin, but in the video we will watch next, you can see that it is quite a distinct community. We're going to follow the development of an infant gut over 27 months. These data are from a study conducted by collaborator Professor Ruth Ley from Cornell University. And we will show you them in a 3D animated visualization produced by Night Lab postdoc Antonio Gonzalez Peña and graduate student Yoshiki Vasquez Baeza. The yellow sphere here illustrates the infant's gut community as it is changing over time, with a yellow path recording the change. We see that the infant gut changes over time, but with a lot of variation from time point to time point. But overall, the community is becoming more similar to that of a healthy adult gut. At one point, the infant was administered antibiotics for an ear infection, and we can see a major regression in the community, but it bounces back really quickly. Finally, after 838 days, we see that the baby's gut microbial community resembles that of a healthy adult. We will learn more about current research on the development of the gut microbiome in an upcoming interview with Dr. Elizabeth Costello at Stanford University. In week 4, we will build on the information you learned during this lecture and the upcoming interviews to learn more about the human gut microbiome and your health.